Well, we're starting genetics, which is my favorite. I'll tell you guys a little bit of background about myself and before I became a teacher tomorrow during class, and give you an idea of why I love genetics so much when I started getting involved with genetics and becoming interested in it. So, when we're talking about living things and we're talking about eukaryotes, prokaryotes, asexually reproducing or sexually reproducing living things, they have sets of characteristics that are inherited from a single parent in asexual reproduction or parents with sexual reproduction. This is called heredity, the delivery of characteristics from parent to offspring, and the study of heredity is called genetics. This is what makes each organism unique. Gregor Mendel was the man who is considered to be the father of genetics. He was an Austrian monk. He was in charge of the garden, the monastery garden, and in that garden, specifically, he studied pea plants. They're small, they're easy to grow. A single plant can produce hundreds of offsprings, and because of that, pretty easy for him to study. It's called a model system because the conclusions that can be drawn there from an easily studied model can be interpreted and the conclusions can be applied to other living things. In the case of genetics, obviously we're more concerned with humans than any other organism. So because there are growing seasons and short lifespans, Mendel could figure a lot of this stuff out with genetics way faster than if he used, obviously, humans or any other large animals. What Mendel knew is that the male part of each flower, around the middle, the male part makes pollen, and that contains sperm. Those are the male reproductive cells. So he knew that. He knew that the female part of the flower, which is this middle part here, made egg cells. He knew that sperm and egg cells come together during fertilization to produce a new cell. In plants, that creates a plant embryo that's encased within a seed. Peas are self-pollinating. Sperm will fertilize eggs within the same flower. So that means there's not a whole lot of mixing and matching. A plant that's grown by self-pollination inherits all of its characteristics from a single plant that bore it. So it has a single parent. There's, like I said, not a whole lot of variety, not a whole lot of mixing and matching. These are called true breeding plants, another way to describe it, a self-pollinator. So, in other words, the traits would be the same in every generation. A trait is any characteristic of an individual. In this case, with pea plants, seed color, plant height, and that might vary from one individual to the next. So what Mendel decided to do was cross his stocks of true breeding plants. He literally took a paintbrush and took pollen from one plant and fertilized another plant. So he basically directed the reproduction and the mixing and matching of traits. This is called cross-pollination. So he cross-pollinated plants with traits different from those of their parents and then studied the results. Specifically, he looked at 7 different traits, each of which had 2 different characteristics. So with seed color, either green or yellow. The offspring of crosses between parents with different traits are called hybrids. Just like a hybrid car that has two different engines, a gas engine and an electric engine, we have two different traits that are being crossed. The original pair of plants that were crossed are P, or parental generation. So that's what you see right here, the top row of the chart. Probably easier to see if I circle it. Or not. Sorry. So these are the characteristics that Mendel looked at. Seed shape, seed color, seed coat, pod shape, pod color, flower position, and plant height. The offspring of the parental generation are called the F1, or first filial generation. You just have to know F1. So the results he saw here were these. So in other words, if you cross a plant with round seeds and a plant with wrinkled seeds together, you end up with the offspring all round seeds, and so on and so forth. So what you see happening is all the offspring had the characteristics of only one of their parents. Round, yellow seeds, gray seed coat, smooth pod shape, green pod color, axial flowers, and tall plants. Whatever the characteristic of the other parent was, looked like it disappeared. It was hidden. Came up with two conclusions from these results. The first conclusion is an individual's characteristics are determined by their parents. Today we call the factors that are passed from parent to offspring genes. Each of these traits was controlled by one gene that had two different varieties, two different forms of each trait. And the two different forms of a gene, or multiple different forms of a gene, are called alleles. Mendel's second conclusion was the principle of dominance. This explained why some of those characteristics of the parents seem to have been hidden away. Some alleles are dominant and others are recessive. An organism that has at least one dominant allele will exhibit that form of the trait. An organism with a recessive allele, that recessive allele will only show up if a dominant allele is not there to overshadow it. So an organism has to have two recessive alleles in order to show that trait. So if we're looking at plant height over here, the allele for tall plants was dominant and short plants was recessive, because tall ended up showing up in the first filial, the F1 generation. Another example here was seed color. The allele for yellow seeds was dominant because it showed up in the F1 generation. The allele for green seeds was recessive. So where'd these recessive alleles go? What Mendel did was another cross. So he took two of the offspring from the F1 generation and crossed them together. And this is what you see happen. All of a sudden, that recessive allele shows back up. Most of the offspring are tall, but about 1/4 of them turned up short. So all of a sudden, that short gene is showing up again. Like I just said, about 1/4 of the F2 plants showed that recessive allele. That dominant allele had masked the corresponding recessive allele in the F1, and then in the F2, shortness had separated from the allele for tallness. How does this segregation occur? Mendel's suggestion was that those alleles segregated from each other during the formation of gametes. So in the F1 plant, each plant got an allele from tallness from the tall parent and an allele for shortness from its short parent. And that's what you see here. When each parent produces gametes, then, the F1 adult produces gametes, the alleles segregate from one another. So you have each gamete carrying only one allele for each gene. That's what you see right here. They separate from each other. We always use a capital letter for a dominant, capital letter for a dominant allele, lowercase letter for recessive. So that's why we have the two different cases there. So now we see the combinations, all the possible combinations here of these gametes in the offspring. So the gametes, if you look here, big T can go with big T. Big T can go with little t. The little t from the left-hand plant can go with the big T. Or the little t and the little t from each plant can go together. So these are new combinations of alleles in the F2 generation.